welcome to our discussion on the cell membrane. So the cell membrane is the outside layer of the cell. It's kind of like the skin of the cell. It's also oftentimes called the plasma membrane. So here's a pretty generic picture of a cell, and again this thick red line around the outside is the cell membrane. The problem with a picture like this is that really the cell membrane, like a cell, is 3D. So three-dimensional. So here's a little bit better representation of the cell and a little closer look at the cell membrane. So starting off with the central part of the cell membrane, the phospholipids. So we talked about phospholipids in our last chapter. They are two fatty acid tails, see those here, these squiggly lines, and a phosphate head. And one of the really cool things about phospholipids is that they're what we call amphipathic, which means that they have a polar and a non-polar region, meaning they're going to both love water and hate water at the same time. So the polar region is the phosphate head. Because the head is polar, it interacts with water. It's called hydrophilic, which means water-loving. If you guys remember back to Unit 1, we talked about how water is a polar molecule. Polar molecules like other polar molecules. So the head loves water, hydrophilic, and the two fatty acid tails, well, they're nonpolar, which makes them what we call hydrophobic, water-fearing. So they don't want to interact with water. So it's kind of a cool dichotomy that these phospholipids have. So phospholipids, imagine them floating around in early Earth oceans, and those hydrophilic heads would want to remain in contact with the water, whereas the tails, the hydrophobic tails, would want to gather away from the water. So they'd form these early structures called micelles. They might look something like this. Here's one that's been cut in half so we can see the tails on the inside. So again, imagine it's surrounded by water, and so all the heads point outward where they can interact with water. And all the tails still stay dry in the middle. And that's called a micelle. If that micelle were to start to grow, it can form a double layer. And that structure is called a liposome. So here's a liposome. And again, here's that double layer. We have heads on the outside, facing the water, and heads on the inside. So this inner cavity here, those heads would also be in contact with water or a water-like substance on the inside of the cell. And the tails, again, stay in the middle, nice and dry, away from the water. So again, this liposome allows for that interior space, which is important for a cell. And this is what we call the phospholipid bilayer. So bi means two, so we have two layers of phospholipids. And with this general structure, we can get cells that are quite large. This phospholipid bilayer can grow, that inner cavity can grow, and you can start to get a lot of material inside that inner cavity. So here's the phospholipid bilayer, heads on top, heads on bottom, tails in the middle where it's dry. So the very first cells that would have formed probably wouldn't count as cells by today's standards. They're probably very little more than a phospholipid bilayer, so we see that here. Back to our two-dimensional drawing so we can see it better. They're easier to make too. Anyway, these primitive cells would have been a phospholipid bilayer, and on the middle, maybe some RNA, probably very little RNA, but just enough for instructions, genetic instructions, genetic code, and maybe a handful of proteins on the inside, but not really what we'd consider a cell today, but just enough to get us started. These early cells, put air quotes on there, they may have lived for thousands of years. And the reason why they may have lived for thousands of years is because it may have taken them thousands of years to build up enough energy in order to replicate and then divide. Alright, so it leads us to our modern day cell membrane. So the structure of the modern cell membrane is to protect the cell and allows what goes in and what doesn't go in. So we can see that here. Some things can go through the cell membrane, other things can't. And it's kind of the cell's job, the cell membrane's job to do that, to determine what gets into the cell and what doesn't get into the cell. Get useful material in, keep harmful material out. So the cell membrane separates the inside of the cell from the outside of the cell, keeps everything inside that needs to stay inside, keeps everything outside that needs to stay outside, and decides what goes through and what doesn't. In general, the whole membrane is formed because of that phospholipid bilayer. So yes, we see some larger proteins in here, but mostly the membrane is comprised of these phospholipid heads on the outside, tails in the middle. So although phospholipids are definitely the primary component, they really only represent about 50 to 80% of the membrane, depending on what else is in the membrane. And so let's take a look at some of the other things we find regularly in a membrane. So in addition to phospholipids, we also have cholesterol in the membrane. So here is cholesterol. You guys remember that ring-like structure that we saw, the carbon rings? Here are cholesterols that are embedded in the membrane. The job of cholesterol in the membrane is to keep it fluid. We don't want these tails getting tangled up and getting too packed together. Otherwise, our fluid membrane would lose its fluidity and become firm. Think of butter. Also, we have proteins, lots of proteins with various jobs these proteins have. So some proteins are receptors that can signal to other cells, or can signal to the cell itself. It interacts with proteins on the top. We kind of talked about some of those receptor cells when we did our quick brief unit when we looked at diabetes. And insulin is a protein that interacts with cellular proteins to help get glucose into the cell. So other proteins are responsible for selective transport. So like a gate through the cell membranes. You kind of see this one here. It's got like this channel here for things to come through or into the cell. Generally, we break membrane proteins into two classes. Integral proteins, which span the entire membrane. So here's an integral protein. You can see it on the top. You can see it on the bottom. There are also peripheral proteins. Now, peripheral means side. If you've ever heard of peripheral vision, it means how well do you see to the side. So peripheral proteins are only on one side or the other. So here's a peripheral protein, here's a peripheral protein, here's a peripheral protein, here's more peripheral proteins. They're only on either the top or the bottom, the inside or the outside of the cell. So integral proteins go all the way through. Peripheral proteins are only on one side or the other. All right, in addition to those proteins, in addition to the carbohydrates, you see them all here in this picture, in addition to the phospholipids, we also have carbohydrates. And hopefully you're recognizing from our last chapter these long chains of hexagons that are coming off the cell. And those are carbohydrates. A lot of times carbohydrates are attached to a protein. So you see that here, in which case they're called a glycoprotein. Again, glyco should sound familiar. Again, we talked a lot about that when we worked with our carbohydrates, and then kind of combining the word glycoprotein, it's a carbohydrate on the protein. Carbohydrates oftentimes work to send signals and act as like feelers on the surface of the cell. Imagine a cell doesn't have eyeballs. It can't see where it's going, so a lot of times those carbohydrate chains are kind of like fingers that are feeling out where it is and interacting with the cell's surroundings, like fingers might in the dark. All right, the components of a cell membrane are always free to move, or not always, but they're often free to move. That's what we call fluid. So here's an example in the membrane itself. Those phospholipids are free to rotate, they're free to move sideways, left and right, front to back. They're pretty constantly moving around. They're not just stuck in one place. They can also even do a flip-flop where they go from one side to the other. Although that's rare, doesn't happen often, it can happen where they flip-flop. They go from the inside to the outside. So here's a little GIF of the fluid mosaic model. It's a lot of different pieces. We've got proteins, we've got carbohydrates, we've got lipids, we've got cholesterol. We've got all those things in the cell membrane, so it looks like a mosaic. And they're all free to move, kind of like ships floating in the ocean. Here's a green protein, here's a yellow protein. They're just kind of floating around in that phospholipid model. And again, we call that the fluid mosaic model. Here's again, another picture of all those pieces, why we call it a mosaic, because there's a lot of things going on.